very creamy.
All right, so we've got Nelly up here. Finny, make sure your body's not blocking the camera. I'm gonna clean her up first.
To make all of those jones, because we don't want to drink the jones or the milk.
Yeah, good job, Finn. So this is just the baby wipe, just to clean up any dirt or dust or mess that's on her.
Or spit.
Alright, so I'm gonna collect the first 3 squirts, which we normally discard.
I'm watching you on here.
Okay.
No, show it up. They can't see them.
So that's about 10 milliliters.
Yeah.
Alright, then I'm gonna milk a little bit into my pail and then I'll collect another sample, but I want to get like mid-milking sample. Alright, so now I'm gonna collect more milk, mid-milking.
Oh. It's okay. Is it okay if I give her a treat because...
So here's like a mid-milking sample, and then we're gonna filter some of the milk I milk in here and collect a sample of that in the house. All right. That's probably enough. Let's save the battery so we can... All done.
Alright, in this video I'm going to be doing some analysis of raw milk. I've got milk straight from the teats where it's normally discarded, so it's flushing the teat out. I have milk that is from the teat, so mid-milking basically, and then post-filtration. I have a control. This is a sell-by date of October 9th, and today is October 4th. And I have a sample of whole milk, so this is 2% milk. And then I have a sample of whole milk that has a sell-by date of 2 days from now. First thing I'll do is I'll just pour some of this 2% milk into a sterile tube. And I'll put this on ice, the rest of the samples. And what I'm going to be doing is diluting the milk into phosphate buffered saline, which will allow any bacteria basically not to lyse in the test tube. Had I used water, there's a potential for cell lysis depending on what's there. I got sterile buffer. These lines on here means that it's been autoclaved and sterilized. So this was sterilized the 26th. September, hasn't been opened. So the first thing is we will be pipetting some phosphate buffered saline into some sterile test tubes. One mil in each tube. I'll also be running a control of this buffer saline on the media that I'm using to ensure that it is sterile. And the goal for this is to get a idea kind of a sample of how many bacteria per milliliter of milk there are in each of the samples. It's an estimate of the bacteria. So what I'm doing is I'm using eosin methylene blue. This is a differential, selective and differential media. We're looking for gram-negative coliform bacteria. So if there was contamination from the animal bedding that got, say, E. coli in the teat duct, this would show that E. coli and other gram-negatives. And then I have a standard plate count media. The only thing that I don't have is typically with milk testing you add powdered skim milk to the media. But again, I'm just trying to get a rough estimate of bacterial numbers in raw milk versus store-bought milk. So there's two main types of pasteurization methods that are used for milk products, both involving heat. There's a high temperature, short period of time pasteurization milk process, and the pasteurization process is just to reduce the number of bacteria in the sample and hopefully kill any harmful pathogenic bacteria like potentially bad E. coli. The high temperature short period of time is 72 degrees Celsius for 15 seconds for milk. And then there's ultra high temperature pasteurization methods, which is 140 degrees Celsius for less than 1 second. So it's heated up really fast and then cooled really fast. And that's typically, if you've ever bought organic milk, that ultra temperature pasteurization method is used, because if you notice on the sell by date, that will last a lot longer in your refrigerator than just a regular pasteurized milk would last. So coffee creamers as well would be ultra pasteurized. But the heat pasteurization method is used to denature proteins. So like heat for canning fruits and vegetables is another method used to kill those harmful bacteria, reduce the number of bacteria, and hopefully, if done properly with home canning, you kill all the bacteria so that there is no food spoilage. When we sterilize test tubes and media, we use what's called an autoclave. That's 121 degrees Celsius with 15 pounds per square inch of pressure. And that's used to kill all bacteria, including what are called endospores. And endospore bacteria are commonly found in soil and can basically survive the canning process if you do home canning, if not done properly and long enough. So for home canning purposes, the endospores can survive the canning process and live in the anaerobic environment of that can and produce toxins. So like Clostridium botulinum produces a toxin, and say if it was like salsa or something like that you would consume cold, that toxin then can be ingested if it was there and can make you sick with botulism. The botulism toxin is not heat stable, so if you reheat that, you can actually denature, kill that toxin. But there are some bacteria like Staphylococcus aureus that can produce heat stable toxins that you consume those even after cooking, and those can make you sick. So very important for home canning to properly follow the guidelines by the USDA to ensure that you are doing the process correctly, so you do not have any of those bad bacteria that can survive in the anaerobic environment. So what I'm going to do is I'm going to start with the control 2% milk. If I can find it right here. And I'm going to put 0.1 mils or 100 microliters into 1 mil of broth, or not broth, phosphate buffered saline. And I've got sterile pipette tips here, if I can open them. These sterile pipette tips have a little filter in them to help keep things aseptic. So I've got this set to 100. actually give this a little shake on our vortex mixer. If you can see that. So that just kind of resuspends everything. 0.1 mils is not a lot of volume. We don't need a lot of volume. We just need to get a sample of the milk. If there's bacteria in high numbers, and we will be able to calculate. There we go. I got 0.1 mils. This is a 1 to 10 dilution. It's been diluted 10 times. Give that a little shake. Clean tip. And I will take 0.1 mils out of this. And put it into a second tube with 10 mils, or 1 mil. Give that a shake. So that's been diluted 100 times. Take 0.1 mils out of there. Give that a little shake. And I'll do it one more time. So that's 1,000 times diluted. And 0.1 mils out of here. And then I will actually discard 0.1 mils out of this. So everything is even. So I've diluted it 10, 100, 1,000, 10,000 times. Do that with the rest of them, and then I will plate these out. I'll show you how I do that. Alright, so I got my control plates here labeled 10 to the minus 4, 10 to the minus 3, 10 to the minus 2, 10 to the minus 1. And then I have my selective and differential media, 10 to the minus 1 and 10 to the minus 2. So what I'm going to do is I'm going to put 0.1 mils on each plate. And the goal is to evenly distribute the bacteria onto the plate. And we want to obtain a plate that has a number of bacteria between 30 and 300 colony forming units is what we refer to them. So a colony forming unit is basically one single cell that was in that sample that forms a colony that's visible, and then we can count it. So between 30 and 300, that's a standard. If it's below 30, we say it's too few to count. If it's above 300, it's too numerous to count. That's just kind of the standard. So I'm going to start with 10 to the minus 4 and work my way to the least dilute, so I don't have to change the pipette tip in between each one. So I'm not increasing any bacteria numbers. So let me know in the comments what you think the results will be. Will the store-bought milk be more contaminated with bacteria, or will the raw unpasteurized milk be more now? Got these sterile, what we call hockey sticks. I got a little pottery wheel here. I'm going to spread the bacteria around, or the milk product around, to get even distribution. And we want to then let that liquid soak in to the media. So again, I can use the same hockey stick here because I'm going from the most dilute to the least. So I'm not increasing any numbers of bacteria. So this one, if there was bacteria, it would have more bacteria than the previous. And this would have more bacteria than the previous, so again, wouldn't be increasing numbers. And since I'm changing media, here I'll use a different hockey stick. -2. So a fun fact about bacteria in the human body, depending on what you read, the body is composed of about 100 trillion cells, and 90% of those cells are bacterial. And, well, I should say microbial, so fungal spores and stuff like that. 90%, so 90 trillion cells are microbial in the human body. So a lot of those are concentrated in the gut. It said about you have about 5 pounds of microbes in your gut system. So I'm going to repeat that process for the rest of the samples and check in with you in a little bit. So my hypothesis would be is that the first squirt milk would have the highest number of bacteria compared to straight from the teat or post-filtration. However, my other hypothesis would be that the post-filtration has the higher number of bacteria compared to any of the other raw milk samples. And that would be because the post-handling procedures of the milk is typically where you would get the contamination. Now this milk was hand milked and not from a milk machine, which milk machines can have problems with biofilm formation in the tubing. But why I'm saying the post-filtration would have higher numbers is because it's not a bacterial filter that is used. And the milk pail is just run through the dishwasher and not sterilized. So what we would find would be sort of normal bacteria that would be commonly found in either from your hands and things like that. So this is the post-filter. I teach microbiology, so I've been doing this for, I think this is my 12th year. So I don't typically do lab experiments anymore. It's probably been, 12 years since I've done an actual research project. Alright, I'm going to save you the boredom of watching me do lab work. Let's film it. Now, regardless of the outcomes of this test, there are risks involved in drinking raw milk, which is why most states don't allow the sales of pasteurized milk. Herd shares in Michigan are legal, which a person can basically own or lease part of the animal and has a right to that portion of milk, depending on the contract that's laid out. I would be very leery of doing any herd share without visiting that person's farm and seeing how they do handle the milk post milking, how they clean their equipment. We show you how we do ours. Our setup is not ideal, but we are as aseptic as possible with what we are working with. all boils down to cleaning the animal's udder good, doing the post-teat dips, and handling the milk properly, getting it cold right away after you are done with the milking process. Checking for mastitis is important. You know, the sale of unpasteurized milk is law in I think most states for a reason, because there can be pathogenic bacteria present in the milk, and people have gotten sick from it. People have died from consuming raw milk. Personally, I am 100% confident that consuming raw milk, when handled properly and done properly, is 100% safe, which is why we drink it. Alright, so I've tried to be as aseptic as possible, closing my pipette tips. I'm not just leaving these little hockey sticks out. They are covered, they were sterilized. Keeping my tubes closed when I'm not using them. Covering my plates right away. Done everything that I can to be as aseptic as possible. And being as unbiased as possible. I've stated my hypothesis that the post-filtration milk is probably going to be the most contaminated with bacteria. And I also expect that there will be more bacteria in the store-bought milk than the fresh raw milk. So post in the comments, let me know what you think. In 24 hours I will check these plates and we'll do some counts and we'll see what the results are. So we'll check back with you tomorrow.